Hi everyone, I'm Matteo. I work at USC. I'm a statistician, and some of my research is about statistical inference for GWAS data. So that's what I'm going to talk about today. I was told this should be a tutorial, so it's not going to go too much into the technical details, but I'm going to give you an overview of this line of work. This is something that with my PhD advisor and several collaborators have been working on for a few years. There are a lot of papers on this topic. I'm going to give you an overview of some of the things that I worked on and what the main themes. So it's about the analysis of GWAS data. So you can think of GWAS data as a data set that collects genetic and phenotype information about a large number of individuals. So think of the UK Biobank. However, since I'm a statistician, I'm going to start by introducing some math notation. And so I'm going to try to make the problem as abstract as possible. The phenotype, and I'm going to focus on one phenotype of interest, is going to be represented as Y. And the genotypes are going to be represented as X. We have a matrix of them. It's a numerical matrix. These are allele counts, so it's going to take values 0, 1, or 2. And there's n individuals in the data set, and there's p variables, p genotypes. It will also be useful at some point to introduce the haplotypes. So the haplotypes basically represent the alleles that are inherited from either the mother or the father. And they're such that the genotype matrix can be seen as the element-wise sum of the two haplotypes. The haplotypes are also written as X, but there's a superscript, M or F, depending on whether it's from the mother or the father. Often, the GWAS data set, the biobank, also collects a lot of other variables that are not genetic, and we're going to refer to this as the covariates Z. To give you an idea of what is the scale of these data sets that we're interested in, the number of variables is very large. For the UK Biobank, for example, it's about 600,000. These are just genotypes that are directly observed through the SNP array. And the sample size is nowadays also quite large. So you can think of it as over the order of 500,000. For some studies, there will only be phenotype information for fewer individuals. There are also biobanks that are larger than this. In general, the question is to try to discover which of these variables, or genotypes or SNPs, are affecting the phenotype in a biologically interested way. However, since I'm not a biologist, I'm going to focus on a statistical interpretation of this question. And so I will try to phrase this question in a way that we can answer using statistics. And then I will describe some of the common themes of the statistical methods that we have developed for analyzing this type of data. And this is not the only possible way to analyze this data. I'll try to relate it to what are some of the more mainstream traditional ways of analyzing this data. So the more traditional approach is to do, well, there are two types of ways actually, two types of statistical ways of formulating the problem. One way is to formulate the problem in terms of marginal associations. So you could ask whether the phenotype Y is independent of xj. So xj represents a particular SNP, a particular variable. Remember that we might have like 500,000 of them. Okay, so in general, we're going to think about this problem not as a biology problem in this talk, but as an hypothesis testing problem. So one way of formulating this hypothesis is in terms of this marginal independence. And so there will be one hypothesis for each of the 500,000 SNPs that we observe. And when you test this hypothesis, this is the standard way of representing the results of these hypothesis tests in terms of p-values on a negative logarithmic scale. So the smaller the p-value, the higher it is on this negative logarithmic scale. And the variables are sorted here based on their physical position on each of the 22 chromosomes. And the SNPs whose p-value is sufficiently small will be higher. And sufficiently here means that the typical significance threshold is basically 5% with a multiplicity correction because we're testing lots of different hypotheses. And so this line here is basically corresponding to 5 times 10 to the minus 8, which is considered a standard threshold for statistical significance. So what you can see from this plot, and this is an analysis of a highly polygenic phenotype using a large data set, what you see is that there are lots of SNPs who pass this threshold. So if you look for SNPs that are marginally associated with the phenotype, for many phenotypes of interest, many complex traits, you will find lots of SNPs that are not independent of the phenotype. Now, why are they associated? Well, there are different reasons why they could be associated. It doesn't necessarily mean that all of these are biologically interesting. In particular, one feature that you can see from this plot, even though the scale doesn't really allow you to see clearly which SNPs are associated, one feature that you can already see here is that there are spikes. There are some areas where there are lots of nearby SNPs that have a strong marginal association with the phenotype. And the reason why that happens is because of linkage disequilibrium. So nearby SNPs on the same chromosome are highly correlated with one another. That's because they tend to be inherited in blocks. And so if you have one SNP in a position, you can predict quite accurately what the nearby SNPs will be. This means that if one of them is associated with a phenotype, the many nearby SNPs will also be associated. It doesn't necessarily mean that any of them or all of them are biologically important for explaining the phenotype. So one way of oversimplifying this discussion is to say that association does not imply causation. That's why this type of marginal analysis is not enough to really gain a lot of insight into which SNPs are biologically interesting for explaining the phenotype. And so that's why there are other types of analysis, often done as a secondary type of analysis, where we try to look a little bit more closely at trying to tease out which of the SNPs are actually important for explaining the phenotype. And so one way you can do that using statistics is by using multivariate models. So instead of trying to test the marginal associations, one can try to build a statistical model, for example, a linear model, to explain the phenotype as a linear combination of multiple SNPs in the same chromosome, in the same sub-area of a chromosome in the same locus. And so this is broadly called fine mapping, this type of linear models that are trying to find which SNPs are important. So linear models typically don't test marginal association, they test some kind of conditional association. And so now I'm going to talk about conditional associations and a different way of testing them that is not through linear models. So the difference between a marginal association and a conditional association is that they test the different types of hypotheses. So the null hypothesis that a SNP J is not conditionally associated with a phenotype is that it's independent, so Xj is independent of Y, given all the other SNPs that are not J, and possibly also given some other covariates. So this is what we call the conditional independence hypothesis. And if you can reject this hypothesis, it actually gives you more information, more detailed information about this SNP being potentially biologically interesting. Okay, so it basically means that if you knew a lot of covariate information about an individual and a lot about their genotypes, also knowing what allele they have for this SNP would give you additional information about the phenotype. So it still generally falls short of making a causal inference about the SNP being biologically important, but it's closer to that. And in this talk, I'm going to talk about two things. So basically, one is how to test this kind of hypothesis, and then I will discuss some situations in which we are actually able to interpret this hypothesis in almost a causal way. So basically, when this can take us closer to finding biologically interesting variants, and what prevents us from doing that in other situations. Now, one thing that I should say, though, is that testing this kind of hypothesis is very difficult. Often you need an extremely large sample size to reject this null hypothesis, and so it's not always practical to tackle the problem this way. Often it's better to slightly relax the problem, and instead of testing each SNP one by one, whether they're conditionally independent with the phenotype, often it's better and more practical to proceed groupwise or at a lower level of resolution. So one way you can do this is to try to group together nearby SNPs that are on the same chromosome and are physically close to each other, especially if they're in high linkage disequilibrium, meaning that they tend to be very similar to each other. And once you group them, and I'm not going to get too into the details of how you group them, but basically you group them based on similarity and physical proximity. Then you can test a slightly modified version of this hypothesis where you test independence with a whole group of SNPs, conditional on all the other groups. So this symbol here, minus G, means all the SNPs outside of a particular group G. And so this is a different type of conditional independence hypothesis, and this hypothesis here is a special case where there's no group in each SNP belongs to its own group. So if you're able to test this hypothesis, and we've written many papers about how you can test this hypothesis for GWAS data, then when you reject one of this, basically we would like to interpret it as saying that there is at least one SNP in this group, for example, G3. We don't really know whether it's X6 or X7, but it could be one of those that has some distinct information about the phenotype that is not captured by the other SNPs. Okay, so that's the type of hypothesis that I'm interested in testing. And again, there's going to be a multiplicity problem. So there are many of these hypotheses that need to be tested because there are hundreds of thousands of SNPs. And so even if we group them in fairly large groups, we might still end up having thousands of hypotheses that need to be simultaneously tested. And so when you test a lot of hypotheses, you need to make sure that you don't make too many false discoveries. And there are different ways of accounting for the multiple testing problem. One way that I briefly mentioned before, the threshold that we saw on the plot before, was trying to control the probability of making a single false discovery. Sometimes that's a little bit too stringent, and it might be enough to make sure that we don't make too many false discoveries. And so a meaningful way to quantify whether we made too many false discoveries or not is to count how many discoveries that we made are false as a proportion of the total number of discoveries. And so that's called the false discovery proportion. And a common goal in multiple testing is to try to control this false discovery proportion, at least in expected value. So that's what the false discovery rate is. And we will see some statistical methods that allow you to test these hypotheses simultaneously, all of them, while ensuring that you should expect no more than a certain fraction of the discoveries you make to be false discoveries. Okay, so basically just by chance they don't actually point to real associations. And so you can think of this false discovery rate as trying to be controlled below 10%, for example. So that would give you some confidence that 90% of the discoveries that we make are not just spurious associations, but they are statistically significant. And so there are two key ideas that are shared by these model X methods that test this hypothesis while controlling the false discovery rate. And so the first idea will tell you why it's called model X. So the first idea is to try to use our knowledge of genetic inheritance and the models that are typically used in statistical genetics to describe the distribution of genotypes in a population to help us test this hypothesis. And so because I said that X is the symbol that represents the genotypes, that's why it's called model X here. It's because we will try to use models for the distribution of X. And these models come from the fact that we can think of the genotypes of an individual as being given by some combination of the genotypes of their parents. In particular, if we could see the haplotypes inherited by the mother and the father, we would see that they are basically a mosaic of the original haplotypes of the parents. So here the mother had a blue and a red haplotype, and she passes on this combination of them to the child, and the parent and the father does the same. So you can think of this biological process as being reasonably well described by a hidden Markov model, a very simple hidden Markov model. And this is being used quite widely for other types of applications. And these model X methods are able to use these models to help us test conditional independence hypothesis. So that's the first key idea, and that's why it's called model X. The second idea is to try to use machine learning models or statistical learning models to detect as powerfully as possible associations from the data. And the way that it's done is by computing importance statistics. So these important statistics can, for example, be given by the coefficients of a linear model. It could potentially be a sparse linear model, or they could be important statistics calculated using some other type of nonlinear model. Here, I'm going to assume that each SNP gets an importance statistics, which could be 0, could be non-zero. It's indexed by j, because j is the index of the SNP. And since we're interested in doing inference at the level of groups of a few SNPs, we also need to combine these importance statistics group-wise. And so there are many ways of combining them. One intuitive way would be, for example, to take the sum of the individual SNP importance statistics and sum them over all the SNPs in a particular group. Here's a plot of what these importance features would look like in a toy simulated example. Here I have simulated a data set with 1,000 SNPs, and I fitted a sparse linear model, the lasso basically, to predict a simulated phenotype using these 1,000 SNPs. And the phenotype depends on some of these SNPs, but only a few of them. And here I'm plotting these importance statistics, and I'm showing in red the importance statistics that correspond to SNPs that actually are used to simulate the phenotype. So those will be the true discoveries that we're after. And the blue ones are the importance statistics for SNPs that are not actually important. So if we reject the conditional independence hypothesis for those SNPs, we're actually making a false discovery. Most of them are 0 because it's a sparse model, so it only gives non-zero importance statistics to a small subset of these SNPs. So now the question is, how do we assess statistical significance here? In practice, we wouldn't see the colors here. We don't know which SNPs are actually important. That's what we're after. And here's the second idea of this Model X framework, is to use negative control variables. That's why we needed to model the distribution of X. The distribution of X is used to generate some synthetic data, some synthetic genotypes, in a careful way, so that they look like the original genotypes, but they don't have any distinct association with the phenotype. Okay, and so that's why they serve as negative controls. And when we do the same type of statistical analysis on these negative control variables, some of them will also show some spurious associations with the phenotype, but they will behave in a way that is statistically indistinguishable from that of the non-important variables in the real data. And so now when you compare these two plots, and remember that we can see in practice these two plots, we just cannot see the color, you can start to infer, at least in a statistical way, which of the variables on the left-hand side plot are likely to be red, because it's going to be the ones that behave significantly different from the variables on the right. So that's how you use these negative control variables to test the conditional independence hypothesis. And the negative control variables are obtained from the genotype data through those models that describe the distribution. So we have worked on different types of techniques to generate these negative control variables and then to assess significance by comparing the importance statistics for the original genotypes and for the simulated negative control genotypes. There are broadly two different lines of work here. One is digital twins, and in the interest of time, I'm probably not going to get into a lot of details, but digital twins basically are only relevant for trio studies. So trio studies you can think of them as being very special types of these genetic association studies, where you have data, both phenotype and genotype data, for both parents of each individual. Okay, so in that case, we can use this hidden Markov model that I talked about before to describe the distribution of the genotypes in the offspring conditional on the genotypes of the parents, on the haplotypes of the parents, because in this case, we actually have the haplotypes of the parents. And so this is actually a pretty accurate model. And what's nice about this is that the model that describes the distribution of the genotypes in the child, given the parents, is a model that isn't really affected by a lot of the confounders that we would typically be worried about in an association study. And so we have a paper where, from a few years ago, where we basically showed that we can use this hidden Markov model to generate some negative control variables and obtain a statistical test that allows us to test this conditional independence hypothesis, where we are able to condition on a lot of confounders that one is typically worried about in association studies. And as a result, we get something that is very close to causal inference. Okay, so there are some downsides to this procedure, though. The main downside being that it requires trio data, which is not so easy to acquire. Most of the time, we have genome-wide association data where we have a population of individuals, but we don't necessarily have information about their parents. We can still test the same condition independence hypothesis, but now we use a different type of negative control variable that is called a knockoff variable. And I don't want to show you the technical definition of the knockoff because it's a little bit too long and I have limited time. But the main idea is that it's still obtained by leveraging these hidden Markov models. The difference, though, now is that the haplotypes that we want to model, they don't come with the haplotypes of the parents. So we cannot directly say that they are a hidden Markov model that is combining the two strains from the parents. Instead, we use a different type of hidden Markov model that has been developed and used for many years for other purposes. The main purpose being phasing. So phasing means that starting from the genotype data that is directly measured by the SNP array, one can reconstruct in a statistical way the haplotype sequences inherited by each parent. So the way that that's done actually is by using hidden Markov models. And this can also be used to impute the missing variables if you have some missing observations. So the main idea of these hidden Markov models is that you model the haplotype sequences in your data set as a mosaic of haplotype sequences from other individuals. And what's nice about it is that this can be done in kind of a recursive way where basically each individual is modeled as a combination of the other individuals. These models are quite accurate, and we have developed a way to use these same models that are typically used for phasing and imputation to actually generate our negative control variables. And what we obtain is, let me show you this plot. Okay, so what we obtain is a synthetic data set that contains this fake haplotype data that is, however, very similar to the real haplotype data. And we're able to do it in a way that not only preserves linkage disequilibrium, so if you compare the associations between different genetic variables and their negative control variables, it's very hard to see any difference. But we're also able to do it in a way that preserves population structure. So here I'm showing you PCA plot for individuals in the UK Biobank, which we can roughly group into these different ancestries. Okay, so most of the individuals will have British ancestry, but we also have smaller subsets of Irish, Indian, Chinese, Caribbean, African, and this is how we define these buckets. Of course, there are more diverse individuals in the data set, but you can roughly divide them into these main buckets. And if you do a PCA plot, you will see basically how genetically related these individuals are across different ancestries. And our knockoffs, so these are basically this fake artificial copies of these individuals, they roughly have the same population structure. Another thing that we can do is that we can try to preserve familial relatedness. So we know that some of these individuals are cousins and parents of each other. And this relatedness can be preserved and reflected accurately also in the fake genetic data that we generate for these individuals. Once we have constructed these negative control variables, we are still able to basically obtain this kind of plot where we calculate the association statistics for all the SNPs in the real data set and for the SNPs in the negative control data set. And then by comparing them, we're able to come up with a significance threshold where we will reject the null hypothesis for all SNPs or all groups of SNPs whose association statistics are above this threshold that we're able to calculate it. And we can calculate this threshold in a way that ensures that the false discovery rate is below whatever level we want, for example, 10%. And so in this toy example, that's what happened here. On the left, we have a plot with association statistics for different SNPs. Some of them are truly important, some of them are only false discoveries. And once we calculate this threshold using what is called a knockoff filter, and that basically is done by comparing the left-hand side plot to the right-hand side plot, then we find that most of the SNPs that have, or groups of SNPs that have a statistic above this threshold are actually truly important in the sense of those condition independence hypotheses. And we can do this for different levels of grouping. And here I'll give you a preview of what the results look like for the UK Biobank. So we did this at scale. We studied several complex phenotypes using basically all of the data in the UK Biobank, all of the genotype data. And this is what the discoveries look like. So this is a simulated phenotype, and I'm showing you a simulated phenotype because here I know what the causal variables are because I use them to generate the phenotype. And basically, here I'm showing a very zoomed in Manhattan plot, basically. So I'm showing like a very small piece of a chromosome. And each genotype SNP here is represented by one dot, okay? And all of this, so here I'm showing you the p-values for a standard marginal association analysis done with what was at the time the state-of-the-art software for doing marginal association analysis. And the important variable here would be this variable here. So this is actually the causal one. But all of these p-values are below the significance threshold. This is something basically where there isn't enough power here. This is a low power study. With our approach, we have a couple of advantages. One of them is that we're controlling the false discovery rate, which is a less stringent statistical criterion. The other approach is that we can use a more powerful multivariate model, in this case the lasso. And what I'm showing you here basically are the shaded blocks correspond to groups of SNPs for which we found a statistically significant association. And we can repeat the analysis at different levels of resolution. So at the lowest level of resolution is the bottom panel. We basically are able to say that there may be something important in this segment that contains several SNPs. When we do a more refined analysis using smaller groups of SNPs, we're able to narrow down the position of the causal variant within this group. There's actually two causal variants here, but we only pick up one. And if we try to do even finer analysis using smaller groups of SNPs, we don't find anything because we lose power. When you increase the sample size, what you get is that we have more precise discoveries. Both the marginal analysis and this conditional analysis are going to find some discoveries. But the difference is that the conditional analysis is able to tell us more precisely that there are two distinct signals here. There are two different SNPs that have a distinct association with the phenotype. Whereas with the marginal analysis, it finds four SNPs that are associated with the phenotype, but they're associated in a marginal way. So it doesn't tell us as much information about how many distinct biological associations there may be here. One final thing I wanted to say, this is actually very recent, well, this is a more recent line of work with one paper that is currently still under review, where we've extended this to do an analysis that is more personalized, in the sense that our interest here is in finding variables that have a conditional association with the phenotype within a particular group of individuals. And basically the idea is that genetic associations may not, like genetic effects, different SNPs may have different genetics associations in different groups of people. And I'll show you one example of what we're able to do. So here we looked at a trait that had already been observed before by several prior studies to show different associations among males and females. So this is the waist to hip ratio. And I'm showing you here the results on the UK Biobank data. So using this type of Model X conditional analysis, we find about 484 distinct groups of SNPs that have an association with this trait. And on top of that, we are able to discover that 46 of them have a distinctive association among females, but we don't find an association among males for those groups of SNPs. And vice versa, there are 11 groups of SNPs scattered across the genome. They're the ones represented in blue, for which we find an association among males, but we do not find an association among females. What's nice about this analysis is that we did not specify in advance whether we thought the SNP would be associated among males or females or everyone. We use what is a simple machine learning model and to discover these associations from the data. So basically, we have a model here that adaptively discovers from the data whether an association appears to be uniform across the population or specific to one sex. And then we're able to test some hypotheses about whether this association is significant or not after we've discovered it using the same data. And in the meantime, we're able to control the rate of false discoveries. So in a way, you could think about this as being somewhat related to looking for interactions. So here will be a genetic interaction with the covariate sex. Although it's not quite the same thing because we're not able to say exactly whether these 46 discoveries are also significant among males. We only have 1/2 of that coin. So basically, we're able to find that they're significant among females, and we just don't have much evidence about they're also significant for males. This is actually consistent, quite consistent with previous studies that found, so there have been several studies of waist-to-hip ratio. They found between 7 and 44 sex-specific loci. So overall, we found a bit more here. There are 57 that we found in this study that appear to be sex-specific. And there's a lot of overlap between the ones that we found and the ones that were found before. So that's one direction in which we've been expanding this Model X analysis of GWAS data is by not only looking for conditional associations, but also trying to look for subgroup-specific conditional associations. In this particular example, the subgroups were defined based on sex, but you could potentially do this analysis using different covariates. In fact, you could throw in a lot of covariates and hope to discover some SNPs that have association among, oh, I'm doing the thing I wasn't supposed to do. You could find some SNPs that are associated maybe among females and some that are associated among people with a certain ancestry and so on. So you could actually throw in a lot of covariates and try to adaptively discover which of them appear to modulate, in a way, the genetic association. In practice, we haven't had a lot of luck with other covariates, probably because we didn't look at the right phenotypes, and there isn't a lot of prior research on looking for this type of more specific associations. But I think it's an area that is interesting, and maybe you have more ideas on what kind of analysis could be interesting here in terms of phenotypes and covariates. So with that, I don't want to use more of my time, so I'll wrap up here. I'd be very happy to discuss this in more detail and give you references and if you want to learn more about this topic. Thank you.